Inside the digestive tract lies an immense physical wall. It is a highly secure border designed to absorb nutrients while continuously defending the bloodstream against microscopic pathogens and toxic waste. This barrier is constructed from a single layer of specialized cells called enterocytes. Because they operate in a harsh environment of digestive enzymes and abrasive food particles, these cells sustain constant damage. To maintain a secure perimeter, the body must replace this entire cellular wall every few days. This relentless cycle of biological construction and cellular turnover requires a staggering, non-stop supply of energy. The gut lining is always hungry, demanding raw resources to power its rapid growth and repair. While many tissues in the human body can operate using glucose, the unique structural demands of the gut wall rely on a more specialized resource. The primary fuel for this high-turnover tissue is the amino acid glutamine. This specific molecule provides the energy required to keep the intestinal grid functional and intact. In this environment, a healthy gut is not a static structure, but a hyperactive economy. It is constantly burning resources to maintain the integrity of its borders. To understand why this fuel is critical, we must look inside a single enterocyte to observe how it processes resources differently than other cells. Through glutaminolysis, the cell breaks glutamine down inside microscopic energy factories, feeding a loop called the TCA cycle. This produces two outputs: ATP to power daily operations, and carbon and nitrogen streams to manufacture DNA and RNA, the instructions to build new cells. When fuel is abundant, the resulting energy activates an internal cellular manager called the AMPK pathway. This manager oversees the physical engineering of the cell's outer defenses. Under this pathway's direction, structural proteins move toward the edge of the cell membrane. They push outward and interlock with neighboring cells to form tight junctions, microscopic protein locks that weld the cells together into a perfectly sealed wall. Creating an impenetrable defense is a matter of uninterrupted energy supply. As long as the fuel flows, the barrier remains closed. However, the local supply for the gut depends on the stability of the entire body. When we face severe physical stress or sickness, the body's energy budget changes. A major crisis forces the body into a hypercatabolic state. In this state, the demand for fuel rises exponentially. The body begins breaking down its own muscle proteins to release stored glutamine into the blood. While the muscles supply more fuel, the immune system becomes an aggressive consumer. Defender cells like lymphocytes and neutrophils sweep through the plasma, rapidly absorbing the glowing glutamine molecules to power their fight against infection. Simultaneously, the liver begins pulling glutamine from the blood to manufacture glucose. This ensures the brain remains fueled during the emergency, but it adds a second major drain on the resource pool. This chart shows the result of this double drain. As immune and liver consumption spike, the circulating supply of glutamine in the blood drops into a deficit called hypoglutaminemia. The supply line to the gut wall is effectively cut off. During systemic stress, the body performs biological triage. It diverts resources away from the structural maintenance of the gut to fund the immediate needs of immune defense. Back at the gut wall, the consequences of this severed supply line become visible at the cellular level. Without incoming fuel, enterocytes experience an internal crisis. This triggers a self-destruct sequence called apoptosis. The shrinking cells neatly fragment into smaller pieces, dying faster than the body can replace them. As energy levels drop, the structural managers inside the remaining cells power down. Without fuel to maintain them, the tight junctions between the cells physically disassemble. The protein locks snap and retract into the membrane. This leaves the once impenetrable wall with unprotected voids between the cells. This allows intact bacteria and toxic waste to leak through these unpowered gaps directly into the bloodstream, a process known as bacterial translocation. This state of leaky gut is a cascade of cellular bankruptcy. The physical barrier fails because the cells no longer have the energy required to hold themselves together. To reverse this collapse, the body must restore its energy balance through supplemental resources that can pull the gut out of its deficit. However, providing standard free glutamine is often ineffective. It is typically absorbed and used by the upper digestive tract before it can reach the damaged cells in the lower intestine. To solve this, researchers use a stable dipeptide called alanine glutamine. Unlike a single amino acid, this paired structure remains bound together as it moves through the digestive tract. This stability allows the fuel to reach a specialized door in the gut membrane called the PEPT-1 transporter. The dipeptide passes through this gateway unbroken, delivering fuel directly to the failing enterocytes. With fuel restored, the cellular managers reactivate. The tight junctions move back into place, welding the gap shut and sealing the barrier completely. This recovery demonstrates that the health of the intestinal barrier is tied to its metabolic supply. When the cells have the specific resources they need, they can maintain the physical seal that protects the rest of the body.